Thank you very much, Claudia, for the kind introduction and also for the invitation to give a presentation today here in this virtual pub meeting or the TechExchange. It's my great pleasure. I'm, as Claudia already said, CEO of TissueGnostics, also co-founder of the company, and overview of our technology platform, our software and application. Very quickly, just 20 minutes. So, if you have not heard of TissueGnostics before, or maybe have not been updated, the company has been founded in April 2003. So just last month, we celebrated our 18th anniversary. The headquarters is in the city of Vienna, in Austria. We have international branch offices in Romania, at the US East Coast, West Coast, and are currently strongly growing in the Asia Pacific area. By last month, we have reached now more than 1,700 reference publications from our clients in more than 60 countries on all continents except Antarctica. What I would also like to mention or emphasize is that TissueGnostics is not just a software company. We are in fact a manufacturer of medical devices and have been certified for ISO 13485 first time in 2008. So our instruments are also in vitro diagnostics compliant. Ever since our foundation, I myself have actually been trained as a cell biologist and worked in cancer research before I founded TissueGnostics. We have been closely linked to academia, and among more than a dozen of Austrian national R&D projects, we have also been involved in 5 Marie Curie innovative training networks on digital pathology, the calcium sensing receptor, the anaplastic lymphoma kinase, cell migration studies, and big data analysis for clinical benefit using machine learning. Our scanners or scanning tissue cytometry platform consists mainly of microscope systems from Carl Zeiss. As you can see, these are high-end microscopes, upright or inverted. TissueGnostics is OEM partner of Carl Zeiss Germany. We have solutions from very straightforward bright field scanners for 2 slides, over systems for bright field and fluorescence scannings, also inverted, where slides and well plates can be analyzed. Also including time kinetic experiments, as well as live cell imaging. For high throughput scanning, we have a slide loader for up to 120 slides, and all our fluorescent configurations can be equipped with a spinning disc confocal for doing whole slide confocal imaging. Like with any slide scanner, the immediate result of the scanning process is a virtual slide. Here we see an example of a prostate tumor region, where we have here in orange the tumor marker. So this is already the prostatic tumor regions. Here we have the normal area, and we can zoom in like in Google Earth, navigate through the sample, zoom in and zoom out. As I said, confocal scanning using a spinning disc confocal is also available. Here we see from one of our clients, Janelia Farm Research Campus in USA, a confocal scan of an entire mouse brain section at 20x. In order to scan the entire mouse brain, we have to acquire 350 fields of view. So the individual rectangles that you see here represent the individual fields of view. Each field of view comes with 4 color channels and a set stack of 21 layers, so main focal plane, 10 above, 10 below. That means 29,400 images or 115 gigabytes of data have been acquired in 1.5 hours. If you only scan the main focal plane, So no set stack, then scanning of the whole mouse brain section in 4 colors takes 8.5 minutes. And again, you can navigate through your virtual slide like in Google Earth. You do not need to take care of individual fields of view. You just deal with your digital sample and zoom in to see the details, even cellular or subcellular details. As good as these images may appear, and they are only an intermediary result for our platform, because what we are actually doing is analysis. And we have here a number of applications in place that will support you to do tissue cytometry. HistoQuest is the solution for identifying single cells and quantifying antibody-mediated staining in immunohistochemistry. TissueQuest does the same thing for immunofluorescence, and our high-end solution StrataQuest can do basically everything what HistoQuest and TissueQuest can do, and more, like contextual tissue cytometry. And this is what I will show you in this presentation. Let's start with the easiest application of identifying markers that are expressed in the nucleus of a cell. Here we see a typical staining of a peroxidase reaction in column. The marker in brown is well known, Ki67, a proliferation marker. And the obvious question here is how many cells are proliferating? In other words, how many blue nuclei are also brown? Now, what people typically do is they either start counting with the finger on the monitor, or they look and guess. Yeah, so we made a survey among more than 200 scientists and asked them, what do you think, how many cells are proliferating here, or how many cells are brown positive? And believe it or not, the answers that we received ranged from 1 to 40%. And this shows the main problem that associated with visual evaluation, that we always get a range of estimations rather than a reproducible measurement. With our platform, we use it's called TissueFAXS, obviously. We use the nuclear marker as a master channel in order to identify the individual cells. What you see here, the yellow contours, show us what the software has automatically identified as a single cell. And then for each nucleus, we measure how much blue and brown, in this case, is present. Of course, it is not limited to blue and brown. You can measure any color. It's about measuring intensities in markers or in color channels, as we say in fluorescence. And what we get is this type of data output. It looks like a scattergram, or people in flow cytometry would call it a dot plot. But what we see here, the individual dots represent individual cells in a tissue section. So and you work with it and interpret it exactly like a dot plot in flow cytometry, just that the data source is a histological section, stained either by means of immunohistochemistry, in this case, or as we will see later on, by immunofluorescence and also multicolor immunofluorescence. Here on the x-axis, we see the intensity of the blue. On the y-axis, the intensity of the brown. This line here is very well known and very important. It's called the cutoff. It is set in reference to an isotype matched negative control, and it determines the specificity of our staining. So all dots below this cutoff will be referred to as negative. What is above these dots, these single cells, will be referred to as positive. This was an example of a marker that is expressed in the nucleus. So there we identify the nucleus and the measure in this area in the other color channel, or channels plural if available. What happens if our molecule is not only or not at all expressed in the nucleus, but in the cytoplasm? We use the same approach. We identify the nucleus, but then look in the surrounding area in the cytoplasm of each nucleus in a so-called ring mask around the nucleus. This we see here. This is an example from skin cancer research. We have two skin biopsies, one taken from normal skin and the other after UV irradiation. The marker here is called COP1, but this is irrelevant. It's about the methodology here. And normally people would look at this and say, okay, this is 1+ positive and this is 2 or maybe 3+ positive. And after discussing, then you say it's 2.5+ positive. With TissueFAXS, you will get an observer independent and reproducible measurement. Here you see the skin biopsy, the normal skin, and this is after UV irradiation. And we see at a glance that the number of positive cells, this is the cutoff that has been set based on the isotype match negative control, and thereby controls the specificity of the staining and shows what is unspecific or what's the level of autofluorescence in the sample. Here we see we have 16.7% of the cells positive, and this increases to 44.7 after UV irradiation. But what we also see is that the positive population shows a shift towards higher values. So here the maximum is around 90. After UV irradiation, this goes up to 130. So what we visually perceive here as brighter is shown by the shift, and what we visually perceive here as more cells that are positive than in the normal skin, this is reflected too by this value of the positive cells that are counted here and not estimated. And the important thing is, whenever you do this analysis on Monday morning, on Wednesday noon, on Friday afternoon, in Vienna, in Lisbon, in New York, wherever, you will get the same and reproducible measurement. One of the most favorite features or most appreciated features around the globe is that you can track individual cells from the virtual slide to data space and back. So we can always double click on a cell in the image, and it will start blinking here in our scattergram. So we know exactly where this cell is located. Following our motto, from image to data, we consider this a step forward and call it the forward connection. It also works the other way around, from data space. So when you make a gate here in the scattergram back to the image, the red cells here, the red contours, are those cells which are in this gate. So you can identify cells with a certain phenotype, so identify a certain subpopulation of cells, but also then visualize where is this subpopulation located in your sample. We have now seen how markers are quantified that are expressed in the nucleus or in the cytoplasm, or maybe both. Now we can look inside the cells and go for intracellular structures, identify dot structures like in fish, fluorescence in situ hybridization, or RNA in situ hybridization, also including RNA scope for specific mRNA detection. Here the number of dots in this circle are assigned to this nucleus and here to that nucleus. So this would be an example of intranuclear detection. There are many other examples like even parasites that are detected or vacuoles, but I don't have the time to go into that detail. As we're working in the era of precision medicine, it becomes more and more important to increase the number of markers that can be analyzed and quantified in parallel at the same time on the same cell population. One of the techniques that is used is multiplexing. So you take a sample, stain it with, say, 4 markers, 4 fluorochromes, scan it, and then remove the staining or bleach it and stain the same sample again with the same fluorochromes, but in the second round, your fluorochromes represent different antibodies. And this, you repeat this cycle a few times so that you have 10, 12, 15, 20 markers, 20 molecules stained on the same tissue section, on the same cell population. This is called multiplexing. And there is a nice paper from Dr. Kim Blenman at Yale University, who has shown in this paper published in 2019 this circular process, including our TissueFAXS system and in particular the StrataQuest software for the analysis. I don't have the time to go into details, but what you get is this type of output where you have multiple markers here, more than 10 on the same cell population, and each dot that you see here represents a single cell in a tissue section. That was multiplexing, yet another technical approach to increase the number of markers that can be quantified at the same time on the same sample is multispectral imaging, where typically a liquid crystal tunable filter, a spectral camera is used that allows to acquire a lambda stack or lambda cube. We have heard about the set stack, which can be used for making 3D reconstruction. In a set stack, we look at the same field of view in different focal positions. In a lambda stack, we look at the same field of view or same sample in the same focus position, but at different spectral intervals. And then by using a mathematical approach called linear or spectral unmixing, by using a reference spectra of all the fluorochromes in use, the system can compute how much from the total emission comes from fluorochrome 1, 2, 3, 4, 5, 6, and so on. Here we see an example of a 5-color staining. The raw data set is rather nasty. It's overexposed, unspecific, structures are unclear. But after the spectral unmixing, it becomes a beautiful, very clear and specific staining that can be analyzed. A relevant example for this spectral technology or using the TissueFax can be found here in this Cell paper from last year, where, as you can see, the immune response against the SARS-CoV-2 virus was quantified in lung tissue of COVID-19 patients by making 7-color stainings and this scanning with our TissueFax spectra and spectral unmixing. Also here, I do not have the time to go into the science, but we know nowadays that the concept of herd immunity in the pandemic does not work. But if this was a game-changing publication when it went online last year in August, because by the time, a year ago, there were still some countries or politicians who expected that waiting for herd immunity would be the right concept to deal with the pandemic. This paper has shown that the SARS-CoV-2 virus tricks our immune system by avoiding the formation of follicular germinal centers, and therefore the concept of herd immunity does not work in COVID-19. A very important part nowadays is also machine learning or deep learning. This is present in our platform. We are also collaborating with 2 different institutes like the Software Competence Center Hagenberg in Upper Austria or the Queensland University of Technology. In fact, I am at the moment in Brisbane in Australia, where I'm a QUT visiting fellow. And we work here on machine learning technologies for, in this case, prostate cancer analysis. But in our products, we also have an algorithm based on deep learning, which identifies nuclei, in particular in lymphatic tissue, where the nuclei are very often densely packed, as you can see here. But still, after deep learning, there is a very good recognition of individual nuclei, even in dense tissue conditions. Machine learning that can be used on the fly by the users, where they select a few categories or a few areas and say, this is class 1, class 2, class 3. And if you do the training and a few minutes later can apply this classifier to the entire tissue sample. And as you can see here, there are basically no limits how many classes you want to recognize. In this area, we have a skin sample and 7 classes identified automatically. We have seen that we can identify multicellular structures like tissue structures, glomeruli, skin, tumor areas, single cells, subcellular structures. Now we can combine this to do what is called contextual tissue cytometry. We see here a colorectal cancer. In red are the tissue infiltrating leukocytes, identify all the single cells, the tumor area in orange, and then can ask different kinds of questions. In this case, the main question was how many T helper cells, so CD4 positive T cells, infiltrate the tumor area and how many are in the tumor stroma. In this catagram, we see only CD4 positive cells. So this is CD4 as an input gate. On the y-axis is the distance in micrometer from the next tumor area. So we have here gates which represent distance zones around the tumor. And in this quadrant statistic, you can see for each zone, like here 1 to 25 micrometer, how many cells are there in total count, in percentage, as well as in density, cells per square millimeter of tissue analyzed. And using this reverse gating feature that I mentioned earlier, we can visualize then here in red all those CD4 positive lymphocytes, which are just outside the tumor, but not further away than 25 micrometer. In orange is the tumor area. In red are those CD4 positive cells. In green are all the other cells that were identified, but are either not CD4 positive or are not in the range of 1 to 25 micrometer, or here 25 to 50 micrometers. So that means we get a very detailed analysis and differential count on the single cell level. And there are, as you can imagine, many more examples. We have, as I said, more than 1,700 reference papers published. Due to time limitations, I could only give you a very short outlook, but I hope that I could show you that with TissueFAXS cytometry, we can support researchers to move from image to data and thereby help to increase the observer independence and the reproducibility. I encourage you to visit our website, where you will also find this list of reference papers in a searchable database. There you can find who has already worked on which molecule, in which tissue, which disease, using a TissueFAXS platform. And with this overview, I hope I could show you that with TissueFAXS you can explore cells, the fundamental unit of life, at the precision that inspires. Thank you very much for your attention.